What you got there?
So this is a solar-powered water fountain.
Welcome. I am Dr. Marjorie Brooks, and we're in my aquatic ecology and biogeochemistry lab in the zoology program at Southern Illinois University. I'm excited to tell you about our research, but before I do that, perhaps I'll tell you a little bit about my background. So I'm the first person in my family to get a bachelor's degree, and I got my bachelor's degree in mathematics and natural sciences. And then I became so interested in natural sciences that I went into reconstructing paleo environments in the archaeology department for my master's degree. The chemistry was so fascinating that I decided for my PhD to get a degree in ecotoxicology in the zoology department at the University of Wyoming. Here at SIU, I have several projects, and I'll tell you a little bit about them. Both of them involve the biogeochemistry of nitrogen and too many nutrients in our environment. We want to have plenty of nutrients available so that we can grow the crops we need and feed people across the world. But sometimes those nutrients make their way into waterways, lakes, and rivers, and then they can start causing problems. One of the biggest problems that we encounter is harmful algal blooms. So in my laboratory, we're studying creative ways that we can discourage harmful algae, in this case, cyanobacteria, blue-green algae, from growing because they produce toxins. We find that they do not flourish well in aerated water. So working with colleagues, we have developed small solar-powered fountains that disrupt the water, keep it from being stagnant, and actually discourage the growth of harmful algae. That's the field component of the study. The laboratory component of those studies is to bring water samples back into the lab and analyze them to see if the nutrients differ and to see if the cell counts are smaller in areas where the water is being aerated. The other large project in our laboratory is part of a team effort, and we're looking at natural processes that remove nitrate fertilizers from big rivers such as the Mississippi, because those fertilizers are going down to the Gulf of Mexico and they're creating a dead zone in the waters off of the coast of Louisiana. And in that dead zone, there's no oxygen for the aquatic life in that area. We know that when the Mississippi River breaches its banks in springtime and floods the adjacent landscape, that landscape, the microbes in that landscape can actually convert that nitrate into nitrogen gas. And that's what this instrumentation does. It actually measures the gas content inside each molecule, each sample of water. My students and I work together on this very hard. And one of the great things about being a professor at Southern Illinois University is that we have the ability to work directly with undergraduate students on research. During the summer, every year I go out to Yellowstone and I study how beaver impact the ecology of small streams. So if you're interested in going out to Yellowstone, I take undergraduates with me every year as field assistants. I'm always happy to have new undergraduates and graduate students working in my lab. So if you want more information, please click on the link below.